every day visit doctors' offices, and some of these people are sent to pathology labs for further investigation and for blood tests and tissue sampling. All these test results are based on the chemistry that is going on inside the samples that is taken from the patients. You know, there are molecules in these samples that their distribution and their ratios can tell us if there are health problems with this patient. One of the most important types of molecules are proteins, like enzymes that they can control life by their function. Mass spectrometry is a technique that is very powerful to study these molecules. But the problem is mass spectrometric techniques need long, complicated sample preparation steps that sometimes the biological information about the sample that is taken from the patient can be lost during these steps. One newly developed method called DESI was invented at Purdue University a couple years ago, and this technique actually doesn't need sample preparation, or it needs minimal sample preparation, meaning that the loss in the information can be avoided. Our lab uses this technique, and it's actually putting the sample of interest in front of a mass spectrometer, and then by delivering the solvent with something like a micro power washer, the molecules from the sample are taken into the mass spectrometer. This technique works really well with small molecules, but with big molecules like proteins, there's a serious problem. As you can see, when the mass of the protein increases from right to left, the sensitivity decreases dramatically. And this is a problem. So my research specifically is to improve this sensitivity and improve this loss in the actual sensitivity of the mass spectrometer. One thing we did was trying out different additives for the solvent of the DESI. And some of these compounds that we used, they showed promising results for improving the sensitivity when we were analyzing the proteins. And most of them, they didn't harm the system like some other compounds that were previously used. So this result is actually telling us that we can use this technique now for proteins in a much more easy way. And we can actually be hopeful that one day doctors can use this technique and take samples from their patients and be able to say if the patient is having a problem by looking at the distribution of proteins throughout the sample that is taken from them, or if they can say if they could be able to remove a tumor completely from a patient that is having cancer without touching the healthy tissues. Thank you.